Okay, hello everyone and welcome to the people that are attending in person and people that are attending online. So I'm very happy to introduce today our speaker, Dr. Tanya Berger-Wolf. So she's a professor of computer science, engineering, electrical and computer engineering, and evolution, ecology, and organismal biology at The Ohio State University, where she's also the director of the Translational Data Analytics Institute. As a computational ecologist, her research is at the unique intersection of computer science, AI, wildlife biology, and social sciences. So Tanya is a member of the U.S. National Academy's Board in Life Sciences, Advisory Committee of the Global Partnership on AI, on Biodiversity, AI on Biodiversity Working Group, and among many others. And she also has received a lot of awards for her research and mentoring, including University of Illinois Scholar, UIC Distinguished Researcher of the Year, U.S. National Science Foundation Career, Association for Women in Science Chicago Innovator, and the UIC Mentor of the Year. She's also the director and co-founder of the AI conservation nonprofit WildMe, which is the home of the Wildbook Project. And she has given hundreds of talks about her work, including at TEDx and UNESCO AI for the Planet. So thanks, Tanya, for being here. We're excited to have you here, and we are looking forward to listen to you.
Thank you, Anna, and thank you, everybody, for inviting to the beautiful city of Boston and Northeastern, and thank you for being in person. And hi, everybody online. Whoever, can you hear me online? Do you know if they can hear? Okay, great. I am going to then remove this. I know there is a way to remove it. Hiding this. Come on. It's there we go. No? Still no? Huh? Oh, you were no. Okay, hopefully. All right. So with that, first order of business, we have to learn how to pronounce this word. So there's going to be a test at the end. So this is the omics of images, Imageomics. So say it with me, Imageomics. No N. Again, Imageomics. Imageomics. Okay, good. So what is it? So, going back to what is the scientific method, and Poincaré's beautiful book where he outlined it, he concludes the book, and this is this thing, and this is what we base today our Western notion of the scientific method, and I highly encourage you to take a look at it. But he concludes the book with the scientific method consists in observation and experiment. If the scientist had an infinity of time at his disposal, it would be sufficient to say to him, look and look carefully. I say it to my students all the time. But since he has no time to look at everything, and above all, to look carefully, and since, again, something I tell my students all the time, it is better not to look at all than to look carelessly, he's forced to make a selection. And so this notion of kind of looking, observing, and deriving scientific insight from it, generating a hypothesis, that's the foundation of science, of Western science today. And scientists for centuries have been learning about nature and understanding nature by looking, right? By looking and recording what they see, recording what they see with increasingly more and more sophisticated tools of looking, and there you are.
Yeah,
that's it. and more sophisticated tools of recording, but there's also Darwin's finches and Jane Goodall looking at the chimpanzees. So what I would argue that technology has it is a read. What I would argue that what technology brings into the scientific process, and particularly computing, is the ability to look at more things more carefully, right? With the invention of the microscope, we were able to look at things we were not able to see before, zooming in down to this molecular and now even the submolecular level. With the invention of binoculars, telescopes, and satellites, we're able to look at the planetary scale, right? Something that humans were not able to look for. And then came genomics, right? And suddenly, not only we're looking at the things that we were not able to see before, but in genomics domain, genomics appended how we study nature. Genomics appended our definition of the species and phenotype and biological traits. In fact, and the biggest, until that point, this direction of going from phenotype, the collection of traits of an organism, to genotype became genotype to phenotype, right? A reverse connection, direction with the invention of, with the start of genomics, with the foundation of genomics. And until today, we define a lot of the phenotype aspect by the genome, whether it's the genome-wide association studies, where we find correlation among the variants of within the gene, and the correlation to phenotype, to traits, to various other aspects of the biological entity we're observing, or even all the way, or understanding evolution from comparative genomics, but even modifying the phenotype by changing the genotype through CRISPR. What I would argue is that recent development of technology allows us to go back to the direction from phenotype to genotype, because it is giving us a lot more ways to look at biology, to look at the natural world in different modalities that humans were not able to use before, whether it's as I mentioned, the satellite and drone scale kind of, not only visual spectrum, the RGB spectrum, but all other kind of spectra, right, that are available to these sensing technologies, on-body sensors that you can put on animals, from accelerometer and GPS, to in situ sensors such as camera traps, motion-activated camera traps, or the acoustic sensors, and even these guys, right? The sensors that all of us carry and can record all kinds of information about the natural world. So with that, scientists are able to look at a lot more things, right? And by far, the largest, most abundant and readily available source of information about the natural world is still images. We're such visual animals. Right. These images today come from the drones and satellites and camera traps, as well as these kinds of things, and the digitization of biological collections of natural history museums. There are millions of images out there. The one platform that records the citizen science observations of nature, iNaturalist, has more than 130 million observations today of almost 500,000 species. By the way, is 500,000 a lot or a little? How many species of animals, plants, fungi, bacteria? Well, let's leave bacteria out. How many species of animals, plants, and fungi are out there, estimated, total? Throw a number, order of magnitude. No?
10 million?
10 million? 100 million?
A billion.
A billion. Let's
You have to define species though, because it's not so clear.
We're not going to get into that, but that level of species, it's not going to change the order of magnitude, no matter which definition you're using. 100 billion, we're settled. About 9 billion. About 8.4 plus minus 1.5 million estimated. Right, and there's 3 recent estimates that all settle around this 9-10 million kind of mark, and that is a shockingly low number. And of those, only about 2 million are described. So 500,000 observations, amazing, right? Because but it also shows bias, right? There's a lot of bias of where these observations are, because if we look at the highest biodiversity areas of the world, almost exactly where we don't have any observations. Come on, right? But so we are able to look at a lot more things with more technology, bias aside. And the current computer vision methods, machine learning methods, in fact, are giving us the scientists the ability to look a lot more carefully at this massive amount of visual information. Today off the shelf, you can take methods for, within an image for detection and localization of objects of interest, putting bounding box around this gravy zebra or African buffalo. You can do species classification. You can even do pose estimation and the stick figure thing. You can do individual ID and the background environmental reconstruction. You can do a lot off the shelf-ish. So we can take all these millions of images from all the sources, find where the animals are, put a bounding box around them, classify the species, and even down to recognizing individual animal, not only zebra, giraffe, whale, or turtle, but Zippy the zebra, Joe the giraffe, Terry the turtle, and Willie the whale. And so that part, the individual ID, is part of our Wild Book platform and the nonprofit Wild Me that was mentioned. Whole other talk for conservation. But great, fantastic. So we can do it, right? Now, we still, all of this, we cannot go from phenotype to genotype in this. We can't even extract the phenotype from using all of these methods. They're very descriptive. Right? We can find the animals, we can get their species. But what we really want to do is not only to be able to say this is the species of animal out of all these, a bird out of all these millions of pictures. We want to be able to say it's this species of bird because it has an orange belly, it has black behind the beak, and it wobbles when it walks, and it opens its wing in this way when it flies. And this is its habit, right? So these are the traits, right? The characteristics of an organism that are describing its physiology, morphology, health, life history, demographic status, and behavior, things like beak color, like the Mendelian, it started with the Mendelian experiment, Mendel's experiment, right, with the peas, the color, the shape of the pea, the pod, the flower, the plant. But it is also things like behavioral traits. Does the bird weave its nest, or does it do something else with it? Does the possum playing dead when it's threatened? It can be at the species level, or it can be at individual level. The stripe pattern of a zebra, which is different from individual to individual, and even changing whether the traits like the gait, while the individual is a healthy versus not healthy individual. The social interactions, the behavioral traits, but also, a pregnant female has a different morphology than a non-pregnant female. The juvenile head to body ratio different than that of an adult. So those are all types of traits. So we need to make traits computable directly from images. And this is Imageomics. This is the new interdisciplinary field focused on understanding biology or organisms and their function and structure of traits and the observable phenotype from images. Notice there is no how, just like genomics wasn't about using this particular sequencing algorithm or anything else. The how we do it today, obviously using machine learning and computer vision. 5 years from now, who knows, right? It could be neurotransmitters directly in our brain, something.
So why the omics part of it?
Omics is the science in general. So omics designates a field in life sciences that is focused on the structure and function from a particular set of type of data. The biological structure and function from, in this case, images. Yeah. It's funded by National Science Foundation, the Harnessing Data Revolution Institute, which is now 2 years old. Yay. And it's a fantastic international team spanning the globe. And the goal, the focus, kind of the approach that we're taking now in the Institute to kickstart the field is, we have all of these image modalities that come from a variety of sources, as I pointed out, including things like 3D CT scans and digital X-rays of organisms, together with a lot of metadata in the form of text, everything from field guides to image captions and species descriptions, geospatial data of the species distribution ranges and the geotag of the image. as well as the molecular data. And we can leverage the fact that biology is the science of structures. This is the science that essentially invented the notion of taxonomy and ontology, right? Classification is a profession within biology. and so we have phylogenies, taxonomies, the trait ontologies. There's a whole field of biontologies. Goes from molecular and protein to anatomic and organismal behavior. And so we can use those structures to constrain directly the architecture of the machine learning models through the process, sort of broadly fitting within the field of knowledge-guided machine learning, KGML. So that the outcome of it is not just descriptive and predictive models, but we can get to at least interpretable aspect because of these traits, and even point out to some discoveries. So sanity check. Let's take it directly as promised. Let's take a biological structure, take a task that we do very well without a biological structure already, species classification, and see if adding biological structures such as phylogeny gives us anything extra. So these are 200 species of minnows, fish that look very, very similar. I could not tell you the difference between those two species or those two species, but we can leverage the fact that presumably species that are phylogenetically close are also shared some of the traits. And we can compare species classification with adding the phylogenetic information, just going one level up in the phylogeny, kind of hierarchically, or not. And the answer is, this is a sanity check, yes. Adding phylogenetic information matters. The species classification has higher accuracy, particularly in the low data regime and the distribution shift. So out of this, like when you have photographs, even for the same species that are from a different museum. These are scans from biological museums, biological natural history museums. Great. Sanity check, check. Now, can we do something even more creative with this? So we can take is there actually a biological signal in the phylogenetic structure that we can observe directly from images? So we can take the phylogenetic tree and restrict it to kind of higher levels of phylogeny and extract the embedding, the features, the embedding from this is the encoder-decoder model for those who are interested in the, and extract the embedding that corresponds to that level of phylogeny. In this case, was 3 classes, right? And we encode it in this image-derived sequence of features. We bring it down to 8 dimensions. And then we advance and do the same thing. So what we call this is the imageome, right? The encoding, the phylogenetic correspondence of the encoding of the image-based features. So not only we can do the downstream tasks better this way, we can actually directly use the decoder part, the generative AI part from the imageome to generate images and ask questions like, if you want to go from this species to this species, from this image to this image, what would be the transformation? And the transformation that we get out of leveraging the imageome actually corresponds to some of the known mutations in the ancestral phylogeny. Yes.
So you do have access to the phylogenetic tree.
Yes.
And that's, do you try to go backwards, like
to generate? This is the next, the next. Yes. So we're doing, but for that you do need the genome, right? Actually, a little bit of the genome. But yes, this is our 1 hour test. This is a fantastic question, and I'll show you some glimpses of that. But we're going not quite there. We were doing the taxonomic keys for those biologists who are familiar with what it is. We're generating taxonomic keys. But yeah, so then you notice here the transition. For example, it's not between the split and fused tail, which is a mutation-based discrete transition. It does show this discrete transition, not a gradual fusing of the tail, which you would get if you were just taking a walk in the embedding space, for example, right? the fin that shifts, so there's two, and then it shifts like directly here. So this is you know the power of actually leveraging domain knowledge in this case, in a good way. And it can suggest some. So what we're doing now here, we're actually generating a whole bunch of synthetic evolution essentially, where we know where we will know the phylogenetic tree and we know the sequence of mutations for the resulting data set, and seeing if we can reconstruct it. So that's all right. So why all of this? Why do we believe that there's something here to even see, to look at more carefully, to see something that humans have not seen until now? First of all, if you ask any biologist or former biologist, how do you define what's an important trait? Because I'm interested in it, right? Because there's no principled way to really define a set of important traits. The reason for it, there are two reasons for it, by the way, well, three. But one of the fundamental reasons is that our hardware, the visual apparatus, is not perfect. In fact, it's hugely constrained. Yes, we're a visual species, but even like this is a paper from almost a year ago that shows that humans do not have enough red-orange acuity to differentiate between different phenotypes, different species of subspecies of particular moth, which, not surprised, didn't evolve for our benefit. They evolved for signaling to their own species, moths, and to birds, their predators, right? So birds, by the way, I can guarantee you have no problem differentiating between those. But the beautiful thing is also machine learning classifier has no problem differentiating between those phenotypes. So the signal is there, just we cannot see it. Our hardware isn't perfect. So let's look at moths and butterflies. So one of the classic examples of mimicry is Heliconias butterflies. So these are these butterflies that are spread throughout of the mostly tropical regions, but they're like very wide range from mid-South America to mid-North America. What is amazing, and this is one species, Heliconias serrato, and this is another species, Heliconias melpomene. And so these guys are all one species. They can breed with each other. There's no problem. They're all the same species and more closely related to each other than anything in the yellow column that are another species and are also more closely related to each other than. But notice they mimic pairs. They mimic each other in melpomenes for the most part because they're evolutionary newer. They're more recent speciation than they're younger as a species than these guys. But they mimic pair for pair, region for region, to gain the same protection because these are not palatable for birds, and birds learned that these guys do not taste great, and in some cases really foul. And these ones are doing this with in some cases with half the genome, right? They've learned the trick. So the question is, what is that the information there that is, sufficient to signal the birds that don't eat me, but still is, different enough for their own species so they don't mate with the wrong ones, right? So first of all, can we differentiate, right? So we can train a classifier, contrastive learning, and ask, the distance in the embedding space. What is the distance between the butterflies of the same species, Erato, melpomene, between butterflies of different species who are not co-mimics, and here are the co-mimics, right? Somewhere in between. So whatever it is, we don't know yet what it is, but whatever it is, the signal is there and it's extractable. This is what essentially this tells us.
Sorry, but this information come from image processing or from
image, just purely from image. Yeah, this is species classification directly from images.
So you have like two different clusters, clearly separate clusters.
Yeah.
And the mimic species, are they tasty to the birds?
They're not as foul as the original ones. So they're gaining the protection without having to invest in any in giving the feedback of the taste. But the thing is, as I mentioned, this did not evolve for our benefit, right? It evolved for birds and butterflies. They must look for different signals, right? And so the question is, the hypothesis, if it is indeed different aspect, different part, different features, different traits for birds and butterflies, then the butterflies should be worse at identifying co-mimics. If I train you, for example, if I just train my species classifier on just on, let's say, on errata, right? There it is, on errata, and I ask it, hey, can you identify who is the mimic, right? So there it is. The mimics are placed very, very close to, and these are not co-mimics of Melpomene. So the mimics are pretty close to the original ones. Without even knowing that this is a mimic, I can train it on errata and just show it a picture of Melpomene and say, you know, which one is it from the errata, and it will correctly identify it as the co-mimic pair. We can do the same experiment, but this is for the human vision. So the hypothesis is that birds will do much better on the task, and butterflies will do much worse on this task. And voila, this is the bird acuity. And notice we actually, even if I show you the original, we cannot tell the difference between the butterfly acuity because theirs is better than ours. And the bird acuity and ours, but this is the butterfly acuity. The models, they're now models of every vision. And so bird acuity, the difference so essentially what this says, and it's confusing, but the punchline is that the butterflies, the mimics look much different to the butterflies than they do to the birds. That's the punchline. So yes, the signal is there. And what we can do now, we can make this classifier and take the embedding for different vision models and take a walk in the embedding space, and for that walk, generate at steps in this walk on the path, generate the images that would correspond, and so do the same kind of transformation, and to do it in every acuity, in every vision model. And so notice that first of all, there is a very smooth transformation, even though the shape is different. So it actually brings in the shape rather than just editing pixel by pixel. This is a rather Melpomene transformation. So we can use that to identify what is actually common to the mimics and what is different What is this minimal thing that is common to the co-mimic pairs? We can do it by anchoring image registration on biologically meaningful parts of the image, not just, here are the coordinates, but here's the split of the Dane, which is common to all butterflies, and they identified by biologists, so we use those landmarks, and then extracting the patches that are common between the co-mimics of different species and comparing it, doing the same GWAS studies, but now because we know the genes for the orange color, for the red color, and so on, and doing the correlations to the to the patches, and so one, we now have the phenotype to genotype, and we have the minimal pairs. So what we're about to do is to print out the minimal, the synthetic versions of the phenotype, the minimal kind of what we think should be good enough for the birds, put it in the bird enclosure on clay on clay backing, and see which ones affect more, which ones are eaten more. So we have the preliminary, experiments, preliminary results, and the answer is, yeah, we can gradually start editing, essentially drawing the isolines of what is good enough for the birds to fool them, right? So this is exciting, and we can do even further. Sorry, did we skip? Yeah, we can do even further and start really asking, like, can we push this towards interpretable AI? Can we really go from this is the species of bird, this species of butterfly, to the because part, because of this traits because of this spots, right? Because of this color. And so, led by Wei-Lun Chao, Herr Chao, this work of modifying the standard transformer models to really produce, the different channels which produce very different salience amounts that are not that general blobs that are salience amount, but the blobs that correspond to, the chest, the head, highlighting, this is, and by the way, the species, I weirdly did not write the species. The blue head, the red chest is what consistently in every field guide will tell you. But these are easy, right? But it also points to the habitat, which is also part of the field guide, where it is, what is it doing, and then all the other things. But it can also point to the butterfly, which traits, which parts of the wing are the signaling wings, and that's what we can now start using for the how the same species recognize it. Why is it this species, right, and not another? And we can do it in the right vision model, which is the butterfly one. Yes.
So how do you work out the number 8, let's say, or how do you construct that?
By lots of experiment.
Okay.
Yeah. And not only on butterflies. So this is, and not only even like you can do it for car models, right, and things like that. Okay. Yeah.
Also-
I sense the question there. Yes.
So how do you make sure that you get different traits? I mean, because so how do you manage to pick up distinct traits?
We're not there yet. It's a great question, except we're not there yet. So we're now, this is kind of the watch this place, because the work is very, very much happening. My Slack is blowing up. Like talked about this in the version that's about to be submitted to the next conference, right? But the really kind of figuring out how do you also, is it better not to embed any kind of domain information in this, or is it better to embed what is discoverable versus what is describable, right? That's the difference, and we can go both ways. And the thing is that computer vision and machine learning models, conferences and venues very much trend to the describable, and the accuracy is measured on how well do you describe what we already know, how well do you recover what we already know. That's not the science metric. The science is what's discoverable, right? But, I joke that I would be, like really the proof that this is really bringing something new is when we discover, when all of this approach will discover a trait that we humans haven't considered as a hypothesis, that actually that biologists then can have a theory of what would be the evolutionary or the adaptation or the adaptive value or the ecological kind of, what is the selection for this, or what is the mechanism for this, right? And something that we haven't paid attention before. So this would be the real measure of, yeah. So with that, and continuing kind of the theme of what humans cannot see, let's look at the individual level. So the fact that, we can identify individual animals from their stripes, spots, wrinkles, notches. So this is all the wild books, even the shape of a whale's fluke or the dorsal fin of a dolphin. There's a lot of value for that, for conservation and the biodiversity monitoring, and a lot of sort of very applied tasks, even though there's a very hard computational problem behind it. But one of the things that when I tell particularly kids that, we can identify individual zebras from photographs, the question I get asked quite often is like, a baby zebra stripe similar to its mom. Fantastic question. This is a great question, except we couldn't answer it until now. Because this is the other constraint that we humans have. Not only our architecture, like the hardware visual hardware is limited and biased, our middleware is also biased because while we're very, very, very good at quantifying similarity of faces, not only human faces, but faces like, oh, the baby looks like its mom, the baby looks like its dad, or this person looks like that celebrity. We can even do it in like certainly monkeys and seals and cats. We can actually like extract enough information out of facial recognition, our own system of facial recognition, to quite often say, oh, that's probably the cub of that animal. We absolutely cannot do this. No amount of training will enable us to do it. Are those two more similar than those two? Nada. Huh? Oh yeah. By the way, none of those are 4 different animals, but are those two more similar to each other than those two? We cannot quantify this. And no amount of training will let us quantify this. I'm curious to hear. But the same algorithm that identifies individual zebras from stripes also points out to the regions of the animal that make them similar and can quantify the similarity. So, hey, let's use this and do straight correlation to genetics. Great idea, except we don't have genetics of zebras in this case, and we're working right now with biologists to get the genetics of a couple of thousand of these guys, gray zebras in Kenya. When we have that, we can do straight up correlation because we have photographs of all of them. But in the meantime, we had a couple of proof of concept results. So working with with photographs of Indian leopards, where we don't have genetic information, but we have the mother cub information. With the way we ask this question there, because we cannot do a correlation, we ask, here's a picture of the animal. Who is the most similar looking animal other than itself in the data set? Here's what we got back. Cub, mother, cub, mother. And only for facial images, because we have them also not only face, but also from sides and everywhere else. It only pulled out the face images. Intriguing, and so by the way, in reverse was also true. Intriguing, amazing, except we don't have enough face images to like really definitively say that there's something else. Okay, well, something going on, but we're waiting. But in the meantime, we don't have to look at faces, right? We can do this kind of pattern similarity on many other things, including eggs. So birds recognize their kin and do kin selection visually, and almost all of them do it visually. In this particular species, which are weaver birds, have doing it completely visually. It's been tested. Biologists, in this case, Mark Harbler from Urbana, they 3D printed eggs and they could change the texture, the smell, they do all kinds of things. Nothing matters as long as the pattern is the same. And the bird will either accept it in her nest or will kick it out. And in fact, when they 3D printed eggs, so, okay, this is one nest or clutch, in fact. This is one clutch. Clutch is the collection of eggs laid by the same bird in one go. Okay, this is one clutch, another, a clutch, and a clutch. All right. And the reason I know this is because there's a nomenclature in naming, because well, I can tell you the difference between these and this. There's no way I could tell you that this is different from this, right? Without any kind of naming here, or that like this is different from this. Nah? But yeah.
Same bird though?
No. But a great question. Yes, we have information of birds laying it over the years, the same one. So that's a great question. Yes. So, so we can now, like, given this and the fact that it's purely visual, we can now start asking questions like, is there a signal? Is there a genetic signal in the pattern? at all. Right, given eggs, can we tell which clutch it's from? The answer is yes, we can classify within about 60% accuracy, which is way better than any baseline you can come up with without anything. And when you separate it between this general pattern and this general pattern, which is, by the way, we could do with like 99% accuracy into two clusters and do it within, it's like 63% accuracy within each one. And it's way better than any baseline, right? So, okay, there's clearly a signal. So is there more similarity between, you know, birds of the same egg, birds of related eggs, birds of within the same clutch than not? And so the answer is yes. When you do this classification and extract the features, look at the embedding. This is birds of the same egg, eggs of the same bird versus the distance between unrelated ones. Sorry, between a bird and everything else, right? This is a clutch within the same clutch, the clutches of the same bird, but not in the same year, and unrelated clutches. And this is related, mother-daughter, non-related, right? And we have images both for blunt and side view, because clearly birds do not care. But more importantly, this is for the human vision. We can translate it to the bird vision. And in fact, the closest one, because they're finches, right? It's a house sparrow is the closest bird model, a vision model. And I know it's subtle, but every statistical measure will tell you that the variance is much lower here than here, right? So what we can do now, so yes, not only yes, there's absolutely a signal. Pattern is heritable, right? This, particularly this, tells us that not only it's specific to each bird and it's preserved over the years, right, to some extent, but it is heritable, mother-daughter, meaning it's a Mendelian trait, something that we did not know before. So we're doing a lot more testing with this, but the fact that it is a Mendelian trait is a discovery. And what we can do now, not with this population, these birds are not there anymore, but what we can do is for a new data set, which we still would need to collect, but it's completely doable. We can now have these clusters of clutch patterns, and we can start this, you know, using the same walk we did with butterflies, transition from one to another, generating an image of an egg across the way and 3D printing it and seeing how far do you need to be from this clutch, from this cluster, until the bird starts rejecting it. Literally drawing the isolines of kin recognition, starting to understand the mechanisms of kin selection, kin recognition in birds purely from images for the first time. Yes.
How do you translate to bird vision? Are you manipulating
the model? Yeah, so there's acuity and spectrum translation. And so people there's a whole subfield, right? The biophysiology and where they understand, and they've done experiments, and you can now get there are in Python models where you say, like, here are the parameters for this species, go. Yeah, I know, amazing, right? Yes. Yeah, so these are like 2 frequency translations. Yes. So yeah, and you can even get it, by the way, these models at what would it look like at different distances, because it's non-linear. So like, yeah, it's amazing, cool, it's really cool. But this is what we should have been doing all along, right? In fact, with butterflies, going back to butterflies, those were RGB images. We're also using UV images, dorsal and so both sides of the wing, right? And also doing it in motion. So we're doing slow motion videos to see, so we can track the trajectory of the orange spot. Maybe it's the trajectory of the frequency, and we actually have the first results is that it's the frequency of opening, closing the wings and the angles. So yeah, so I mean, it may not be just purely that. So there's a lot more fun stuff to do, which we can do purely from images, right? And look at a lot more data, a lot more carefully than we've been doing until now to really drive it to scientific discovery. And we can even go the answer to every AI researcher today to everything is the foundation model. Yes, we have a foundation model. We've trained a foundation model for species classification on about 10 million images from a variety of sources, and in fact, we can now train it in about a week. We've added a lot more. This is a multimodal foundation model, so it's a modification of CLIP, if you care, led by you, Sue, faculty, and we downstream tasks, things like species classification, individual ID, rare species, zero shot, few shot. It performs better than standard, but you can also, we're starting now to in a limited way, let our biologists play with it, because they're coming up with tests just like GPT users, right, that we haven't even thought about. And so it's exciting to see, how quickly they can break it. So the model, but it also means, and we're starting to augment it, because they're like, oh, but we want to see this. Okay, great. Well, let's do this, right? So, but we really want to drive it to the point where it can tell us this is this species of bird because, and the because will include something like, wait, what? We didn't think of it. Oh yeah, but then you have to look at it in UV and shift the spectrum and whatever. Makes total sense. It's bird vision. Yeah. So yes, but this model we're about to also release not only the kind of the first version with the model with it's trained based phylogeny and a lot of other information. So you'll be able to play with this as well, hopefully soon. But as I just mentioned, the signal is not only in the static images, but particular behavioral traits are, of course, the signal is in video, right? And we need video, and the structures that we would be using are these like super complicated things that are called ethograms. I get lost somewhere around here, right? But David here can tell you a lot more about it.
I wrote a code for one on a Palm Pilot, and that'll date me, I think, a little bit. Collect
Yes.
enchiladas. So.
Right, and the first videos of those were recorded using GoPro 3.
Yeah, go great.
Yeah, but so the current version and the project is part of the experiential introduction to Imageomics course, which we teach as part of the institute for students. It's a year-long course with, I'm wrapping up, year-long course, given in this case 8 universities, including UK and Europe, US and Kenya, and the faculty for 5, giving introduction to both computer science and ecology over the fall semester, and then we take students to Kenya for 3 weeks. They work there on interdisciplinary collaborative projects that neither of them could do. Things this year include, and then finish off over the spring, and for some of them it becomes their PhD. Things this year included things like the evolution of toepaths of geckos, and using computer vision, and that's the picture taken from a drone. But the biggest project was extracting behavioral traits from drone videos of zebras, giraffes, baboons. I mean, any species, ideally, but in general, this is so this is a start. So we do have the extracting automatically mini scenes, sort of focusing on one animal, and then automatically identifying individuals and classifying their behavior. So there it is, walking, head up, and so on. And I'm going to skip the rest of this video. The large project still ongoing, lots of annotation that had to happen. We compared it to scientist observation standing in the field. It is very reliable for the observable behaviors, but then we can go a lot further from the drone. And we can even start extracting the because part. This is this behavior because, so the different salience maps. And the exciting thing that we were pushing towards is not only individual behavior, but like group behavior, social interaction, and even interspecies interaction. Notice the displacement there of the zebras coming, and the antelopes are like, oh, okay, fine. But notice this is the fun part, and I'll leave you with this one. Right, so this is something that we would love to be able, and you can do it only from the drone, because you can kind of have the field of view. So there's a lot of work on the system side that we need to do, but clearly, this is an opportunity that is just the potential level to look at more things more carefully. And of course, it's not only limited to wild animals, but lab animals and plants. And particularly big in agriculture, extracting phenotype from images, not only on the organismal, but also cellular level. This is a cancer cell because, right, would be fantastic to have, and the whole landscape level. And with this, thank you very much. And remember how to say the word Imageomics. Yay, okay, great. And I see there are tons of questions there on the, should I monitor myself?
If you would like to, or we can read them out loud.
Yeah, let's do one each. Let's read one from online. Sorry, huh? Oh, wait, what? Oh. Okay, let's okay, so I've answered that already. Yes, so video analysis. We're gonna consider that one answered. The Imageomics approach. What cases where image-based signal is not enough to differentiate? For example, for dolphins, vision combined with echolocation. So this is a great question, particularly for birds, right? For species, behavior quite often is defined through vocalization, through sound. And this is true for other species as well. And in fact, one of the projects currently is just starting on the acoustic side. So clearly vision is not enough for everything. But even from vision, purely from images, right? We're nowhere near of extracting everything that we can. So it would be great. And we're also, I didn't even show the 3D imaging part, but we're working with Morphoscope, Source in Seattle, a project that does 3D imaging and scanning of organisms, especially from museum collections. So there you can go down to skulls and so understand really the evolutionary and the traits and how go way, way back in time. One from the audience will alternate. Yeah.
So I have 2 questions. I can come back to a second one later, but so the first one is, I mean, if you can detect these Mendelian traits, as you mentioned, you could potentially take a picture of I guess I don't know, I guess a school of fish or a flock or anyway.
The school, yes.
Yeah, and you could potentially get the familial relationships, right? Like
in principle, yeah. We
So
hope to get there eventually, yes.
construct a full genealogy.
So yeah, so this is, fish might be hard. We don't know yet. Recognizing individuals and kind of figuring out what are individual traits versus species level traits. But certainly for because we need to be able to verify. So for animals where the biometrics are a good signal, like the zebras or the leopards, or we're in conversation now with the so we will verify that for zebras, whether the stripe pattern is a good proxy for genetic relatedness. Once we have the data, there is an ocelot. Sorry, I keep moving around. Hi online. But we're in conversation with another group of researchers who study a species in Latin America that they do have both images and genetics, and there we can, because if we can do it, it's fantastic. You can start studying questions of dispersal and gene flow in the population and in the range within the species range without having to do this invasive thing of taking their blood or any other way of taking DNA, which quite often you have to tranquilize, capture, and it's a mess, right? If you can just use images, it opens up amazing opportunities for scientists to look at more things more carefully. But it also points to something, where to this day, scientists are arguing and trying to figure out the function of stripes, right? Why do zebras have stripes? We haven't settled on the answer to this question, and it's probably three. And the reason, there are three reasons, fundamental three reasons, three functionalities, is probably because they serve simultaneous to different species, right? The dazzle effect, which presumably makes it harder for cats and predators, is based on the cat vision, right? It has nothing to do with like our ability to see and not to see. And cats have very different vision from us. They don't have color vision, right? They're also the thermoregulation, but for that you need certain width of stripes. And then there's flies evidently don't like striped things or like them less than. So these are three very different like modalities, and it could be, all of the above, but we've never looked in three different modalities, right? So for the first time, to me, the exciting part is, yes, we can start using as a tool, as a proxy for other more invasive ways of studying nature, but we can also discover things that we've never been able to see. The online question. So we're trying to identify jaguar individuals against the database of already identified ones. Oh, this is great. Please talk to me, Regina, because and use Whiskerbook, whiskerbook.org. So it's one of the wild books, or if you go to WildMe and look at the platforms, you can find Whiskerbook, and there are already jaguars in there. So yes, and totally can match individuals. It's easier to match when you're matching to a database of known ones. The hard problem that we're also solving is to be able to say this is a new individual, the open world problem, as it's known in computer vision. So it's not a matching problem. We don't have a reference database. In fact, majority of individuals that we see in any species have never, the majority of the queries when you have an image is, this is a new one, we haven't seen it, because majority of individuals have only one or two pictures. So that means that majority of the queries are going to be new ones. And you have to be able to say that definitively. And so that is a harder query. But certainly, if you're collecting, Regina, let's stop, because you're collecting exactly the type of genetic information that will be able to then correlate, right? So you can do, transfer learning. You can do a lot of things. I would love to talk about this. So for data collection, they use fecal samples right now for zebras. They have blood samples for these cats, for ocelots. They're not cats, for ocelots in Latin America. So different genetic information, but certainly pictures and picture modalities are different. So the answer to this question is, what approaches do we use for data collection when we don't have enough data? As I said, we're solving the open world problem. So the point is to establish whether and which part of the visual data provides a good proxy for the genetic relatedness, and from there you can then start dealing with the missing data.
Okay, Tanya, just for the sake of time, maybe just one more question.
Yes. Question from the audience.
Yeah, so can you tell us more about how the model could change, how the foundational model could change if more users are going to start using it, or like what are your hopes or predictions?
So right now, how the question is how the foundation model would change if more people are using it. Just like when they trained GPT-3 and put it out with a chat interface for people to start using, nobody could imagine the type of queries that people were throwing at it, and so then they trained it up with more data, And now with visual data, right? So and there are many other kind of this encoder decoder based on foundation model with downstream task kind of combinations. We came up with some baseline, downstream tasks and measured the performance based on those. But we're already, if you open it up to biologists, they're going to ask questions we haven't thought yet about. And we'll realize that, oh, we should ingest all of that information to be able to answer this question. Let's put it in. Maybe we should, do more annotation instead of just, more modalities. Maybe we should look at more because there's out of distribution shift quite often. Maybe we should train it on synthetic images, not only on real images. So I think the next version is going to be very, very much driven by the type of questions that biologists will start asking, but they're already starting to ask generative questions. Can you generate the phylogenetic tree that corresponds to this subset of images I'm going to show you? Or can you show me what the back in time images would have looked like if this were the, for the museum collection? Or can you tell me, generate the taxonomic keys, which is if you like try to identify species, for example, it will tell you, oh, does it have, butterfly versus moth, right? Does it have hair on its antenna and doesn't? Okay, if it does, it's moth. Now, does it have this color, that color? Okay, so these are the kinds of things that we want to be able to generate. Anyway.
Okay, so thank you everyone for your questions. I think we need to wrap up now. Thank you, Tanya, again, for sharing this amazing and beautiful research. We are looking forward to see how all of this progresses. And that's it. Thank you.
To answer, sorry,